Hello, everybody. My name is Fazila Mohammed Prem Nawaz, and my topic is on mitigating metastasis and utilizing cancer associated fibroblasts to stop the spread of cancer. Cancer is the most important and common disease diagnosed worldwide. When cancer spreads from one organ to another, it becomes significantly harder to treat, and the health outcome of the patient decreases. This process is known as metastasis and is the primary cause of mortality in most cancers. Metastatic process involves a series of events, which can be shown in the image to the right. These events include proliferation, where cancer cells multiply nonstop from the stroma and form a primary tumor site. It ultimately involves intravasation of cancer cells into the circulation, where they travel as CTCs to sites of metastasis. After excavating out of the blood vessels, proliferation causes a second tumor to form in a new metastatic site. Any failure to complete one single step of the metastatic cascade would lead to arrest of the entire process. This therefore raises hope for finding drugs that prevents metastatic cascade by targeting and isolating specific features of the metastatic cells. In 1989, Dr. Page had proposed a seed and soil theory, which emphasizes the concept of tumor spread. When he said that when the plant grows to seed, its seeds are carried in all different directions, but they can only live and grow if they fall in a congenial soil. Similarly, during metastasis, malignant cells spread from the primary tumor site through the blood vessels of the lymph system to other sites of the body, where the malignant cells from a new tumors override the normal tissue function. In this case, the seed is the malignant cells and the congenial soil is the tumor microenvironment, also known as TME. The idea behind circulating CAVs and CTC clusters, cancer tumor cells clusters, is that cancer cells take their soil with them to make it easier to seed a new metastatic site by supporting the tumor growth. This can be shown in the image to the right, where the cell leaves the primary tumor site and they travel through the circulatory system to seed different metastatic tumors, and they return back to the primary tumor site. The tight interactions between the TMV and the cancer cells form a protective niche that favors tumor formation. The TME is mainly composed of multicellular cells from mesenchymal, endothelial, immune cells, and fibroblasts arranged in the ECM, or the extracellular matrix. Because metastasis presents a serious barrier to effective treatment and recovery, it is crucial to understand how CAFs play a role in the breast cancer metastasis and progression. They are activated fibroblasts and a major component found in the ECM, and they secrete signaling molecules called cytokines and support the invasion, migration, and metastasis. They are often found in the circulatory system along with CTCs. In breast cancer microenvironment, non-cancer cells, predominantly CAFs, crosstalk with cancer cells to induce many of these events. It is discovered that CAFs circulate in patients' blood in co-clusters with CTCs. Using vitro models, this research focuses on signaling crosstalk mechanism, underlining CAF-CTC intravasation, facilitating clustering, and driving role of clusters in the metastatic setting. The purpose of the study was to investigate this physical interaction between CAFs, CTCs, and endothelial cells, an important first step in this process of extravasation, wherein a cancer cell leaves the circulation to initiate a new metastatic site. My hypothesis is that if I target specific proteins in the ACM that help the functions of CAFs and CSC clusters in the blood circulation, then there is a chance to reduce the binding of CAF to the endothelial cells and eventually prevent metastasis. While this work focuses on the relationship between CAFs and cancer metastasis, other proteins in the ACM impacts the CAF's productivity on a wide variety of cancer function, which means that the scope of CAF's impact on cancer is much larger than that just of metastasis. This project was done remotely at home due to the arising COVID-19 pandemic. To replicate the model of CAF-BC, cluster extravasation endothelial binding assay was done. The endothelial lawn was made with a layer of HUX cells. The cultured cells created tight lawn wherein other cells cannot penetrate. CAF-BC cells were labeled with cell tracker dye and pipetted on top of the endothelial layer and incubated for after washing off, pictures were taken. My main project consisted of differentiating the endothelial and the calf BC cells and counting the different fluorescent cells using the ImageA app. Shown in the image to the right, I used the ImageA toolbar from the Fiji app to keep track of the cells. In each experiment, the cells were treated with different specific proteins and media. The first experiment results with a Higgs treatment, which causes HA, hyaluronic acid degrade, which partially inhibits CAF cells binding to the endothelial cells. This experiment is shown to prove this theory. The mock cells are the CAF without Hayes treatment, and the Hayes indicates CAF cells with Hayes treatment. In the image, it indicates that the percent of treated cell bound to the endothelial cells was significantly less than the mock cells. The oscillatory shear stress presents the flow of blood in the blood vessels. The shear stress affects the calf binding to the endothelial cells, which can be represented in the graph where it shows comparison of cancer cells in placed onto the rotating platform at 100 RPM at 0 minutes and after 5 minutes. The percent of calf cells bound right after taking it out of the rotating platform was significantly less than when it was tested after 5 24-hour pre-treatment and the calf media during binding assay shows that the average percent of MD231s, which is a more aggressive breast cancer line, and the MCF7 which is a less aggressive breast cancer line, bound in the endothelial cell in the presence of regular media of calf conditioned media. Both type of cancer cells showed insignificant changes because the treatment did not affect or have much or any effect on the calf bindings of the cancer cells. Possible sources of error I encountered was interpreting the images. In the scanned image to the right, it shows the variety of sized fluorescent dots. However, it was difficult to differentiate which cell would be the endothelial and which cell would be the calf pixie cells. This confusion might have caused some differentiation in the present and the mock cells. To avoid this, I compared my results with my peer who was an undergraduate in UFM college to make sure I had similar results. Another challenge I uncovered was the learning curve. In the beginning of the project, it was hard to interpret the meaning of the graphs and the cell percentage. However, after looking at variety of graphs, I overcame this challenge. The findings reported in the study supported my hypothesis that calf CT clusters in the circulation promote the extravasation of CT cells through their physical interaction with endothelial cells. To sum it up, the calves support the cell growth, cancer cell growth. This research provides a new perspective in the intersection between calves and ECM proteins and other cancer metastases. Because 90% of cancer deaths are associated with cancer metastases, it provides a significant obstacle to effective treatment. Thus, it is crucial to understand how factors from the tumor microenvironment influences way cancer spreads. Additionally, the project focuses on ECM protein hyaluronan, which is an important from this intersection because it supports and increases the calf's number. The blood flow also impacts the binding of calf cells. In the future, I hope to conduct more trials to validate and improve my findings from the experiment. It is also important to study how calves from a variety of cancer lines affect change and affect one another. And furthermore, I hope to evaluate the connection between the calves and the ECM proteins, how it influences the expression of other proteins like collagen. I hope to also explore mechanics of binding and study the role of comorbidities in each of these functions. This project could not have been done without the help of many people. I gratefully acknowledge my mentor, Dr. Angela Sparts, a research associate in the UFM, to offer her support and expertise and give me the chance to explore the world of cancer this past year. I appreciate her encouragement when I presented to the El Ashuri lab meeting and her continued advice. I also want to acknowledge Ms. Geneva Taylor, a biochemistry undergraduate at the Lang Lab in the University of Minnesota, for working with me to analyze the cultured capsules. I'm thankful for Ms. Princessa Hansen, my teacher, for her continued guidance over the year for my research endeavors. Her advice and feedback helped me to grow as a science communicator. Lastly, thanks to my parents for supporting me in trying everything until I found what I love in science. These are the references I used to develop my study, and they were key in how I approached my methods. All images by author. I'm really proud of the work I was able to complete in the study. Thwarting cancer metastasis is critical in addressing cancer therapies. These steps are needed for scientists and doctors to find better ways to disrupt the cancer pathways, to find more effective treatments and better options for patients and their families. Thank you for listening for my presentation.